process of analyzing a substance to determine its composition or quality. The term is often used in the mining industry to refer to tests of ore or minerals. The term assay is also used in the environmental, chemical, and pharmaceutical industries. An assay is an investigative analytic procedure in laboratory medicine, mining, pharmacology, environmental biology, and molecular biology for qualitatively assessing or quantitatively measuring the presence, amount, functional activity of a target entity. The purpose of assay development and screening in drug discovery. Assays are investigative procedures that qualitatively assess a compound or examine a compound's effect on identified molecular, cellular, or biochemical targets. In order to give reliable, usable data, the assay must be robust and reproducible. The assay should be robust in that it is not unduly affected by changes in sample preparation and handling. Equally, it should give the same results regardless of the individual operating the assay.
Chemical assay. Chemical assay is the analysis of sample material known as the analyte using a set of chemical procedures. Chemical assay is essential for regulatory compliance as well as determining the quality and composition of chemical substances and materials used in products, industrial processes, and manufacturing.
There are two types of chemical assay, qualitative and quantitative. Qualitative assay identifies the presence of chemical but does not measure the quality of chemical. Extraction, distillation, precipitation, and other methods that determine the physiochemical properties of the substance. Quantitative assay. This type of assay measures the quantity of the chemical. It is the volume or weight of the substance. To make it simple, qualitative assay in chemistry gives the presence or absence of different chemical components in a sample, whereas quantitative assay gives the amount of different chemical components present on a given sample. These are the physiochemical assay techniques: photometry, colorimetry, spectrophotometry, colorimetry, flame photometry, chromatography, column chromatography, paper chromatography, thin layer chromatography, gas chromatography, and high-performance liquid chromatography.
What is immunoassays? An immunoassay is a diagnostic test that uses the reaction of an antibody to an antigen to diagnose a disease. When a patient is infected with a pathogen, the pathogen can be detected directly by measuring the presence of antigens. Immunoassay is a highly selective bioanalytical method that measures the presence or concentration of analytes ranging from small molecules to macromolecules in a solution through the use of an antibody or an antigen as biorecognition agent. In life science research, immunoassays are used in the study of biological systems by tracking different proteins, hormones, and antibodies. In industry, immunoassays are used to detect contaminants in food and water, and in quality control to monitor specific molecules used during product processing. An immunoassay capitalizes on the specificity of the antibody-antigen binding found naturally in the immune system. Antibodies made by the adaptive immune response in the body are highly specific toward particular antigens. For example, this is why we receive vaccinations, to help our immune system build an antibody repertoire response towards parts of an antigen before we encounter it in a more pathogenic state. The immunoassay will use those highly specific antibodies to probe for molecules of interest when they are in mixture with other molecules. Immunoassays can be used to test for the presence of a specific antibody or specific antigen in blood or other fluids. When immunoassays are used to test for the presence of an antibody in a blood or fluid sample, the test contains the specific antigen as part of the detection system. Immunoassays test works by using antibodies to detect small biological substances in the blood and other bodily fluids. The method follows the aspect that particular antigens bind to specific newly introduced antibodies, thereby stimulating an immune response. 5 types of immunoassays. Number 1. Radioimmunoassay. The radioimmunoassay is perhaps the oldest types of immunoassays. Here, a radioisotope is attached to an antigen of interest and bound with its complementary antibody. Then a sample with the antigen to be measured is added. It competes with the radioactive antigen, kicks it out of the binding spot, and replaces it. After washing away unbound antigens, the radioactivity of the sample is measured. The amount of radioactive signal is inversely related to the amount of target antigen. The health hazards of using radioactive substances cause a movement towards safer methods. Immunoassay. In a counting immunoassay, polystyrene beads are coated with many antibodies complementary to the target antigen. During incubation, the beads bind to multiple antigens and group together into a large mass. Some beads remain unbound. The entire solution is passed through a cell counter and only the unbound beads are counted. The number of unbound beads is inversely proportional to the amount of antigen. Number 3. Enzyme-linked immunosorbent assay. In an enzyme-linked immunosorbent assay, the antibody is linked to an enzyme. After incubation with the antigen, the unbound antibody is washed away. The bound antibody enzyme attached to the target antigen is observed by adding a substrate to the solution. The enzyme catalyzes a chemical reaction of the substrate to produce a quantifiable color change. A practical example is a magneto-enzyme-linked immunosorbent assay system for the detection of CD4 plus cells for the diagnosis of AIDS. Number 4. Fluoroimmunoassay. In a fluoroimmunoassay, the antibodies are labeled with fluorescent probes. After incubation with antigen, the antibody-antigen complexes are isolated and the fluorescent intensity is measured. Number 5. Chemiluminescence immunoassay. The principles of chemiluminescence immunoassay are the same as an enzyme-linked immunosorbent assay or a fluoroimmunoassay, but the reporter is different. Luminescence is the release of light due to an electron being kicked up to a higher energy state of emitting a photon as it relaxes down. This is the same principle as fluorescence. The difference lies in the mechanism of kicking the electron up to a higher energy in the first place. Fluorescence, this is achieved with certain frequencies of light. In chemiluminescence, this is achieved by a chemical reaction. These reactions require an emitter and a co-reactant. A magneto-actuated chemiluminescence assay was developed to detect the presence of Zika virus in patient samples.
Microbiological assays are used during production to determine the potency and quality control. These are used to determine the pharmacokinetics of drugs in animal and human. There are 3 main methods of microbiological assay by which the potency of sample and standard solution may be compared. Number 1. Dilution methods. Number 2. Turbidimetric and tetrametric and gravimetric method. And number 3. Diffusion methods. Microbiological assays are called dilution assays. Dilution is the process of decreasing the concentration of a solute in a solution, usually simplified by mixing with more solvent like adding more water to the solution. To dilute a solution means to add more solvent without the addition of more solute. In turbidimetric assay, particle-bound antibodies are mixed with the sample in appropriate buffer. This mixture is then analyzed using a light source. Level of light transmitted reflects the amount of antigen in the solution. If no antigen are present, the light is hardly absorbed. This diffusion test or diffusion methods is a culture-based microbiological assay used in diagnostic and drug discovery laboratories. In diagnostic labs, the assay is used to determine the susceptibility of bacteria isolated from a patient's infection to clinically approved antibiotics.
Bioassay. A bioassay is an analytical method to determine the concentration or potency of a substance by its effect on living animals or plants, living cells or tissues. A bioassay can be either quantile or quantitative, direct or indirect. The importance of bioassay is to provide valuable information concerning the potency of biological products. This is essential for evaluating batch-to-batch consistency and stability. Bioassay data are crucial at all stages in the development of biological products, from early research work to fine quality control of finished products. There are types of bioassays. First is direct assays. Second is indirect assays based upon quantitative responses. Third is indirect assays based upon quantile responses. One well-known example of a bioassay is the canary in the coal mine experiment. To provide advanced warning of dangerous level of methane in the air, miners would take methane-sensitive canaries into coal miners. If the canary died due to build-up of methane, the miners would leave the area as quickly as possible.